Okay, so welcome to your next genetics lesson. This is section 1.2, part 5. So to begin with, we're just going to talk through the answers to page 38 and 39 in your notes on codominance. Okay, so let's talk through a couple of these examples. They're already filled in for you, so just check over your answers as you go. So a black FB feathered chicken is crossed with a white FW feathered chicken. Identify the phenotypic ratio and the genotypic ratio. Because it's two homozygous parents being crossed, it means that each parent is donating one, so FB and one FW, to each offspring. This means that every single offspring will be FB FW, which is that checkered patterning. So 100% checkered, 100% FB FW. So again, this parent donates this allele, and this parent donates this allele, or it could be that this one donates here, FB and FW to here. Okay, so 100% checkered, 100% FB, FW. Okay, second example, a black FB feathered chicken is crossed with a black and white FB/FW feathered chicken. So notice here that it doesn't mention checkered, for example, it just says black and white. So be aware that this does indicate, although it is stated here in the question, that this is FB/FW. We've got our two parents. So parent 1 is here, parent 2 is here. So one is homozygous, one is the checkered. So if we look here, we've got two FB/FB offspring and two FB/FW checkered offspring. So if it helps, you can do either genotype or phenotype first. I prefer doing genotype, but 50% will be black, 50% will be checkered. Genotypically, 50% will be FB/FB, 50% will be FB/FW. Okay, so this third example is very similar to the black example, except now we're crossing with a white chicken. So a black and white, so again checkered, FB/FW feathered chicken is crossed with a white FW/FW chicken. So we've got our two parents, and again, we have two FB/FW chickens, so 50% checkered, 50% FB/FW, so that's the same. It's just one's the phenotype, one's the genotype. And then we have 50% FW/FW, meaning that we have 50% white. Question 4, a black and white FB/FW feathered chicken is crossed with a black and white FB/FW chicken. So both parents are FB/FW genotype. So if we cross those, we end up with one FB/FB, 50%, which are these two right here, are FB/FW, and 25% are FW/FW. So phenotypically, we have 1 in 4 that's black, 50%, so 2 out of 4 are checkered, 25% are white. Okay, so number 1, is it possible for two black chickens to produce anything other than black chickens? Why or why not? No. If they only have FB alleles present, all offspring will be FB/FB, meaning that they're all black. Number 2, is it possible for two white chickens to produce anything other than white chickens? Why or why not? Nope, this is the same answer as number 1, except this time they only have FW alleles present, so they can only donate those to their offspring. So they'll only produce white chickens. Question 3, is it possible for two black and white chickens, so that means FB/FW genotype, to produce anything other than black and white chickens? Why or why not? Yes, some will be black and some will be checkered and some will be white. So this is related to the Punnett square, the fourth one on page 38. For question 4, what phenotype does each parent need to be to produce only black and white offspring? So again, this is the FBFW chicken. So to have all checkered offspring, one parent needs to be black, one parent needs to be white. So this is the same as the first example on page 38, but essentially if we have FB crossing with FW, you get FB, FW, FB, FW, and so on. These two will be the exact same as those two. So why do we refer to this as codominance? So both alleles are expressed if present. So the prefix co essentially means that both things are valued equally. So think co-workers or to cooperate. So this is why we refer to it as codominance. Okay, so let's get into our actual lesson for today, which is on the second type, which is incomplete dominance, or notebook page 35. So an alternate to codominance is what is called incomplete dominance. So when one allele is not completely expressed over another, i.e. they blend together, this is called incomplete dominance. So instead of being patchy or showing both, this shows a middle ground. One capital is given to the gene, i.e. C for color, with different subscripts for each letter, allele, i.e. R for red, W for white. So really similar notation and setup to codominance, but gives a different end result. So on occasion, you may come across using two different letters for two different alleles, i.e. R for red, W for white. This was used to give a way to distinguish from codominance, but usually this is used in older documents. So we're going to stick with the notation that I just explained to you, okay? Okay, so let's practice this in an example. In carnations, the color red, R, is incompletely dominant over white, which is W. The hybrid color is pink. So here are our flowers. So our notation here is C capital R and C capital W. So then when we do the genotypes for the homozygous dominant versus the heterozygous versus the homozygous recessive, it would be CR, 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 CW, and CW, CW. Okay, so for example, give the phenotypic and genotypic ratio from a cross between two pink flowers. So we are crossing CR, CW with CR, CW. So to start off, we're going to draw out our Punnett square. Okay, and then we'll write one parent is CR, CW. The second is the same. So the first box is going to be CR from the top parent and CR from the side parent. And then the second box on top is CR from the side parent, CW. I'm writing it still in order of what would be dominant. So CR is dominant to CW. The bottom set, CR, CW again, and then the last one will be CW, CW. So genotypically, this means that 25% are CR, CR, 50%, because we have two of them, are CR, CW, so the pink, and then the last 25% RCW, CW. Sorry about the different sizing. It's, for some reason, PowerPoint changes the thickness on its own. Okay, so then phenotypically this means that we have 25% red, that's our dominant, 50% pink, and 25% white. Okay, second example, give the genotypic and phenotypic ratio from a cross between a red flower and a pink flower. So our red flower is CR, and our pink flower is CR, So our Punnett square for this, the parents are already in place, but in the first box, we get CR from this parent, CR from that parent, CR from the top parent, CR from the side parent, CR from the top parent, CW from the side parent, CR from this top parent allele, and then CW from the side. This means that genotypically we have 50% CR, so the dominant, to 50% CR, CW, and that's just from reading. We've got 2 CR, CR on top, 2 CR, CW on the bottom. So phenotypically we have 50% red to 50% pink. So this slide just shows other examples of incomplete dominance. So this can be seen in mice in the trait of fur color. So if we say have FB for black, so FB is black, and FW would be the white. 2 black alleles would give you a black mouse, 2 white alleles would give you a white fur-colored mouse, and the FB-FW mix would give you a gray. This also applies to hair texture. So the curly allele would be HC, and the straight hair allele would be HS. So the incomplete dominance of if you have one allele from each, you would then have wavy hair versus curly or straight hair. So this is the end of our lesson for part 5 of 1.2. Please work through the incomplete dominance practice on page 41 and 42. They are quite similar to the last set of practice, but you want to think about what the end result might be since it's no longer speckled, it's now a combination. All examples as a bonus though are based off of the carnation flowers, so red, white, and pink flowers.